writing the tests, so I'm going to talk about what I think is important, and I'm going to put that important stuff on the test. So that's kind of a way that you can judge whether you're paying attention in the right places, right? Another question that I got asked that I want to make sure we're clear on, you may use lab notebooks for lab quizzes. You may not use lab notebooks for lab exams. So quizzes, the 5 pointers that are just about every day of lab, you can use your lab notebook for, right? Because the point is, are you coming to class prepared with a lab notebook? And the way I check that, because I don't grade your lab notebooks, is give you a lab quiz. And you can pretty much assume that every day you come to class, you're gonna have a lab quiz, and if you don't, then you can be like, yay, I didn't have a 5-point lab quiz today. But you don't wanna be like, oh, there's a 5-point lab quiz today, and I haven't even looked at the lab, right? Because then there's 5 points gone forever. So, prokaryotic cells, right? That is what we care about. Not that eukaryotes are not important, right? Obviously, we're made of eukaryotic cells. So they are important, but that's what we talk about in general biology. So in this class, we're focusing on the prokaryotes, right? And we started way out at the tippy tips, right? We didn't even talk about yet what's inside the cell. We talked first about morphology and arrangement, right? So like this cell right here is a single bacillus, as is the E. coli that you're going to be looking at in lab today. So one of the things that we're going to be doing in lab, it's kind of wonky, right? Because lab 4 is kind of a continuation of lab 2. It teaches you a new technique, how to put bacteria on a slide with a stain and how to look at them. But instead of just doing that, we are going to look at all of the E. coli that have been growing since you put them into different media on Thursday, because we want to see how good you are at aseptic technique. And so when you take each of those 4, right, you've got 2 broths and you've got 2 slants, so each one is going to go on an individual slide, and you're going to put it on there, heat fix it, stain it with a stain called methylene blue to make them blue, right, so that they're more stand-outing than they normally would be, right? Think about your protozoans that you looked at on the first day. They were all colored, right, so that they were easier to see. So we're going to color them to make them easier to see, and we're going to look for the presence of single bacilli, because E. coli are single bacilli. If you don't see E. coli, you killed them. If you see E. coli and something that's not single bacilli, you successfully transfer E. coli and also his hitchhiking buddies from your hands or from the air or something like that, right? So your options, right, you have a couple options. Number 1, single bacilli only. Oops. That is indicative of a good aseptic transfer because you see only what you're supposed to see. Option number 2 is single bacilli and some other morphology or arrangement. So maybe you see single cocci or streptobacilli. But you also see single bacilli, right? So in this case, you say, okay, I did successfully transfer E. coli, but I contaminated him because I have something that E. coli can never be. E. coli can never be strepto, it can never be diplo, it can never be cocci, because morphology and arrangement are a matter of genetics. Just like I'm never going to be 6 feet tall, right? It's a matter of genetics. The other option is no single bacilli and other stuff. And in that case, you did a double no-no. You killed your E. coli and you contaminated it. Right? You're like, how did I do that? Probably your loop was too hot when you transferred it, and maybe you left it open too long so stuff came from the air. Right? A really common contaminant is cocci, because guess what kind of bacteria grow on your skin? Cocci. So it's very common for things like staphylococcus or micrococcus, which are both cocci, to end up in your tubes. And what that says is you were not being careful with your transfer, you were not aseptic. So it's kind of wonky, right? We're going to be finishing up one lab as part of another lab. And all I can say is welcome to microbiology, right? Because that's what we do a lot, is one lab continues another concept, right? So that's what we're doing today, right? So with those single bacilli. So we talked about arrangement. We talked about morphology. We talked about extracellular stuff, right? Like The glycocalyx, which may be really highly organized, like this capsule here, or it might be kind of loosely organized, in which case we call it a slime layer. We talked about pili, right, which are tunnels through which DNA is sent from one cell to another. We talked about fimbriae, which are like bacterial Velcro and help them to attach. And then we talked about flagella, which, just like eukaryotes, are organs of motion, right? They're for motility, but they're different in that we have different arrangements of flagella in microorganisms in prokaryotes, and we also have a different kind of flagellum, right? Remember, it's a rigid structure that spins like a corkscrew rather than being this flippy thing that's flexible like eukaryotes. So some stuff similar to, but each distinct, and that's why we're talking about it in microbiology, right? So finally today, we're gonna move in. But before we get to the plasma membrane, we're going to talk about the cell wall, which is pictured here in sort of a rust color. Cell wall are found in plants, in fungi, and in bacteria. And they serve the same purpose in all three, right? Any cell on the planet is constantly pushing against the desire of water to get inside, right? Water wants to dilute things. So if you have a high concentration of solutes, which you do in every cell on the planet, what does water always want to do? It always wants to get into the cell. So you have this constant pressure coming in, we call that osmotic pressure. And what it will do is make cells pop, right? So you think to yourself, well, how do I withstand that? Well, your cells are little babies that are all perfectly cared for in an extracellular environment that is exactly identical to what goes on inside the cell. Whatever's outside, your blood, interstitial fluid, lymph, all the body fluids are exactly the same osmotic count as what goes on inside the cell. So if water wants to go inside, other water will want to go outside, and your cells stay fine, right? Which is why if you ever get an IV, right, it's like 0.85% saline. Why? So that it doesn't rush, right? So that you don't have damage to your cells because water wants to move. So cell walls are always used to handle water wanting to rush in, right? What will happen if water rushes in? The cell will pop. So cell walls are rigid structures that prevent cells from popping because prokaryotes are all unicellular. Every single one of them lives in an environment that your cell would consider extreme, right? Your cells get their temperature regulated, they get the pH regulated, they get the salt concentration regulated, everything about them, right? They're bathed in this beautiful fluid that is absolutely perfect, right? Which we call homeostasis, right? That's one of the things that we do. Is it's really important for us to maintain that environment. They are unicellular, they can't do that. Now they can, right? We learned they're motile. So what do you think they do if the pH changes and they don't like it anymore? They can swim away, right? What do they do when they find things they don't like? They tumble, right? So they tumble out of the way of a pH that ranges here or ranges here. What do they do if it gets too salty? They can tumble and move out of the way if it gets too hot, if it gets too cold, right? But they're also a teeny tiny cell floating in a big lake or a big ocean or your big body, right? Or your colon, where things are constantly changing. And so their best defense against that is to have a cell wall that maintains their shape, right, and keeps things from rushing in, right? So cell walls prevent osmotic lysis, right? So they keep the cell from blowing up. Osmotic lysis means lysis or splitting, right? Like I said, we're going to talk about glycolysis on Thursday. Glycolysis, glucose lysis, glucose splitting. That's what glycolysis means. So lysis means to lyse, to pop, to split. So when we say osmotic lysis, we mean to pop because of osmotic stress, because so much water rushes in just like a water balloon, right? Put too much water in a water balloon, what's the water balloon gonna do? It's gonna pop, right? Because it can't resist that pressure forever. So the cell wall is not unique, right? Just like flagella, the cell wall is not unique to prokaryotes, but the kind of cell wall that we're gonna talk about is unique to prokaryotes. Now it turns out we can't say much about all prokaryotic cell walls because there are lots and lots and lots and lots and lots of prokaryotes. But what we can say is that the cell wall of a prokaryote is usually peptidoglycan. Right? So if you're all prokaryotes and you all have cell walls, and I ask each one of you what's your cell wall made of, the majority of you would say peptidoglycan. So that is unique, right? So fungi have a cell wall made of something called chitin. Plants have a cell wall made of something called cellulose. Prokaryotes typically have a cell wall made of something called peptidoglycan, which, as its name suggests, you're like, what his name doesn't suggest anything to me, Krista, right? But peptide and glyco means proteins and sugars. So this is sort of a brick wall that surrounds the cell, and the bricks are made of sugar, and the cement between the bricks are made of proteins. Right? So it's a rigid cell wall that surrounds these cells, right? So this is kind of what it looks like, right? So here's the bricks, and here's the cement or the mortar, right? Here are the sugars, here are the proteins, and together they make this very strong, very rigid structure that resists osmotic lysis, right? It helps cells to live in a constantly changing environment, right? Now notice I said usually, right? So there are some exceptions to that. One exception would be to not have a cell wall. There are bacteria that don't have a cell wall. Organisms who are members of the genus, notice it's not italicized. This is just a general name. We call them mycoplasmas, but they actually have, so like Mycobacterium pneumoniae, right? Or Mycoplasma pneumoniae. Mycoplasmas happen to be bacteria that don't have a cell wall. So what do? Well, they do the kind of the same thing you do. They increase the amount of sterols, like your cholesterol, in your cell membrane to make them a little bit more rigid. They also tend to be intracellular pathogens. They tend to be bacteria who live inside other cells. Like Mycoplasma pneumoniae, guess what it causes? Pneumonia. And guess what it likes to live in? Lung cells, right? So that way, it sits inside a perfectly regulated environment. So we have a lot of intracellular bacteria who don't have cell walls, and the way that they survive is because they sneak into other cells, right? As soon as they can, they get inside another cell because now somebody else has to do the work of making everything beautiful for them, right? The other option happens in a big group of prokaryotes that is very unknown, not very well known, and that is a group of prokaryotes called the archaeans, which we're going to talk about in our diversity unit. And they, even though they look, and people thought for a long time that they were bacteria, they actually grouped them all together in Kingdom Monera. They were like, there's all the little guys with no nucleus, right? Let's throw them all together. But when they started studying them, they thought, it's really weird that they never have peptidoglycan. They always have this other thing called pseudomurein. So it turns out, even though they look very similar, they're not very similar at all. So if there's a cell wall, it's usually peptidoglycan. There are exceptions, right? There's exceptions to everything, right? So the exceptions are you can lack a cell wall, or you can have something other than peptidoglycan in your cell wall if you're an archaean, right? But for the most part, it's peptidoglycan, so that's where we're going to focus our attention, right? Especially because this cell wall becomes a very important structure in identifying unknown bacteria, because it turns out that while most bacteria have peptidoglycan in their cell wall, they have a sort of a continuum of concentration. There are some bacteria that have a ton of peptidoglycan and some that have very little. Remember that guy Hans Christian Graham? And he thought he was a total failure, and he's like, well, hopefully someday somebody can do something with this, right? And it turns out somebody did. They learned that his technique of staining bacteria would take all bacteria in existence and put them in gram-positive and gram-negative as a category. Now, where do you think the wall-less bacteria would end up? Negative, right? Because they don't have any peptidoglycan. Where would all the archaeans show up? Negative, because they don't have peptidoglycan. So this gram stain From the 1800s, they had no idea, right? Because think about, could you tell how much peptidoglycan bacteria has in its cell wall by looking at them with your microscope? I mean, you've seen them, right? You've seen cells. Imagine something, 20 times smaller than that, and you trying to see the structure of the cell wall, right? It's not going to happen. So in the 1800s, he didn't know why. He just knew this thing happened, and at the end of it, something was purple and something was pink, and he said, this is positive and this is negative, and what we know today is positive means enough peptidoglycan to put you in the one category. Negative means not enough peptidoglycan to put you into that category. So we have gram-positive, which have lots of peptidoglycan, and we have gram-negative, which have little to no, right? But there's really this spectrum, right? It kind of runs all the way. So over here we would say gram-positive, over here we would say gram-negative, and what we're talking about is the concentration of peptidoglycan. But imagine you can have a cell here, or here, or here, or here. So there's kind of some gray area in there. Now, thankfully for you, we're nice when we make you do this. We give you cells that are on either end of the spectrum so that they're really easy to see. But we want to keep in mind that there's this continuum, right? There's this spectrum of concentration of peptidoglycan. Some have a ton and some have a little. So when we look at organisms who have a ton, right, we can look at a picture of their cell wall. Here's tons of peptidoglycan. We can look at a scanning electron micrograph. Notice scanning electron micrograph, I get that nice three-dimensional picture. I'm very close to it. It's very magnified, so it's not a light micrograph, right? I get a scanning electron micrograph, and if I look at them using a transmission electron microscope, because I can get so close, up to 100,000 times bigger, I can actually see the structure of that cell, right? I can see this cell right here. Notice, right, this guy is a member of at least a diplo arrangement. Because notice, he's completely separate from, but still attached to a cell next to him. Right, so not close to each other, actually attached to one another. He has this very thick layer of peptidoglycan, which is shown here, kind of a dark gray. And then his plasma membrane is inside of that. So very thick peptidoglycan allows you to, right, you imagine when you stain a cell, right, the stain is on the outside and it soaks into the cell. So it's got to soak all the way through all that peptidoglycan. And then you try to rinse it out. And if you only rinse a little bit, you're not going to rinse it out, right, because you have that big, thick layer. It's kind of like, right, so my son likes to be helpful, and sometimes he likes to do the dishes, and when he does the dishes, he will put a huge amount of liquid dish soap into a sponge. Like, it's like a bad dream. It's just frothing, and there's just soap everywhere, right? And it would take me an hour to rinse all of the soap out of the sponge, right? Because the sponge is really good at holding on. That's like that's like that, right? That really thick layer of peptidoglycan really holds on to any stain you use, so it's really hard to wash it out. And so what happens, and when you do this gram stain technique, they look like these guys over here, they're purple. And we go, that's gram-positive. Now I recognize you don't know yet what the gram stain procedure is. You will, and you'll get to practice it on Thursday, right? Your ubiquity broths that you either rubbed on your bods or rubbed on some surface in the lab are growing in a test tube right now. And we're gonna take them out and we're gonna gram stain them. Because they're not single bacilli, who knows what they are, right? Anything that grew anywhere that you swabbed. So we're gonna do a gram stain on those. So you're not even gonna look at those broths today, except to look at them and go, ooh yeah, something grew. And then you're gonna put it away and you're gonna look at it on Thursday. But when we look at it on Thursday, not only will we be able to see single bacilli, we will be able to say, I see gram-negative bacilli, and I see gram-positive cocci. So it allows us to learn more about the organisms by that staining technique, and the reason it works is because of that cell wall made of peptidoglycan. So these are the guys that are way over here, right, on this end of the spectrum, and we call them gram-positive, right? Because even though Hans Christian Graham thought he sucked, we don't think he does, so we call it a Graham stain, right? So we always remember that even though he thought he didn't do anything phenomenal, he actually did something really phenomenal, named it after him, right? What about those guys that are on the other end, right? So what does their cell wall look like? They have an itty bitty bitty, or absent, bless you, peptidoglycan layer. So they actually have two cell membranes, right? Because you think, well, osmotic lysis, right? If I don't have a lot of peptidoglycan, water's going to want to rush in. I'm going to maybe pop. So what can I do? Outside of this peptidoglycan, every one of these bacteria has what's called an outer membrane. We call it a lipopolysaccharide outer membrane. Right? So how did they get away with not having a lot of peptidoglycan? They have a lipo, right, L-polysaccharide, LPS outer membrane. Now, Graham had no idea that that existed, right? We learned that later. What he did was apply that same staining technique that we'll talk about later on, right? Add the purple dye, rinse out the purple dye, add the pink dye, see what happens. And these guys, because rather than being like a sponge, it's kind of like a paper towel or a washcloth. It's really easy to wash the soap out of a washcloth as compared to a sponge. It's really easy to wash that purple dye out of this thin layer of peptidoglycan. And so we have to restain them a different color and they end up pink. So gram-negative cells end up pink because of this thin layer of peptidoglycan. And he didn't know that, which is why he thought he was a failure. Also, there were no antibiotics back then, right? I think it was you who was asking, right? Why do we care about antibiotics? So now you can imagine if an antibiotic targets the cell wall, like say penicillin, if you don't have a cell wall, is that antibiotic going to kill you? No cell wall, no target, right? It's the reason you can take penicillin and it doesn't hurt you. You don't have a cell wall. So anything that damages the cell wall is not going to damage you, right? We call that selective toxicity. But it also is not going to bother these guys because they don't have much cell wall. They don't really rely on the cell wall. They don't need it, right? So it doesn't do anything for them. But these guys, right, if we look at them, right, a cross section, notice that same peptidoglycan, and then here's this itty bitty line. That's their peptidoglycan, just a little bit. But outside of that, we see this outer membrane, this lipopolysaccharide outer membrane, and because it's a membrane and not a brick wall, the cell actually looks kind of ripply on a scanning electron micrograph, right? As opposed to these look really smooth, they look really ripply, right? It's like looking at a brick wall or a hedge as a perimeter around someone's yard, right? The hedge is really bumpy, right? Because it's living, it's organic, it's moving, right? So the cell membrane outside there doesn't have that really nice smooth structure. But they don't mind, because actually that outer membrane is what typically makes gram-negatives more pathogenic than gram-positives, right? Pound for pound, the gram-negative cells that you encounter that could make you sick, a lot of the reasons they can make you sick is that outer membrane. The outer membrane has endotoxins in it, which can cause fever. It has different things in it that inhibit your phagocytes from eating them, or kills phagocytes. There's all kind of interesting things that we're going to talk about in chapter 15, 20, don't remember. Somewhere at the very end, right? So this, right now though, cell wall, right? Most of the time peptidoglycan, it turns out some cells have a lot of peptidoglycan, we call them gram-positive, right? And some cells have very little peptidoglycan, and we call them gram-negative, right? So Graham, if he were alive today, would probably be a happier man because he would realize what a huge contribution he made to the field of bacteriology, right? So one of the most important things that you will ever do in a microbiology class is the Graham stain that you're going to practice on Thursday, right? That's the fundamental tool that a microbiologist uses to identify an unknown bacterium, is Graham stain it. And because we have to let bacteria grow, it's one of the first and most important things that we can tell a clinician when they have a sick patient. Because it takes 24 to 48 hours for bacteria to typically grow from a person's specimen. But you can do a gram stain in just a few minutes and at least tell the clinician what they have in terms of gram reaction so they can start antibiotics if they need to, right? So that's an important consideration, and it can be done very quickly. So you're gonna practice that on Thursday, and you're gonna be using that technique throughout the whole semester, learning how to differentiate bacteria based on the amount of peptidoglycan in their cell wall. All right. Another exception, because there are always exceptions, is to have a really wonky cell wall that still has a fair amount of peptidoglycan, still has lots of peptidoglycan. So would it stain gram positive or gram negative? Lots of peptidoglycan, so it's going to stain positive. But here's the kicker. It has this weird stuff, right? Incidentally, the reason that I put these pictures up here instead of the ones from your textbook is because your textbook doesn't have a picture of this kind of cell wall, and I wanted you to be able to see them, right, in sort of the same structural setup. So here's that thick layer of peptidoglycan, and outside of that is not a lipopolysaccharide outer membrane. It's a mycolic acid layer, right? So yes, we're adding more terminology, right? So mycolic acid is this molecule right here. It's a real jerk. It is what makes things like Mycobacterium tuberculosis resistant to lots and lots of antibiotics, right? Not because it has learned antibiotic resistance, but because that mycolic acid layer is a really waxy film that's nearly impenetrable. It's really hard to penetrate that mycolic acid. It's kind of like, right, it's waxy like a candle. So imagine, right, have you ever gone down, like, you know, I had kids, so we do field trips, which is fun because it's the only time you ever in your own hometown do things that are touristy. Is I've been all over the place, right? So I've been to Old Town like 17 times, and one of my kids, like, it doesn't matter how many times they did it, they always want to dip candles, right? You ever dip a candle, you get a white candle,